joining today, and thank you for tuning in. I am Al-Khalaf, a research fellow here at the University of Ottawa Heart Institute, and I am back as your host for today's episode of BEaTS Research Radio, a YouTube channel and podcast produced by the Bioengineering and Therapeutic Solutions Lab at the Ottawa Heart Institute. Our goal at BEaTS Research Radio is to communicate science to the community. Our guest today is Dr. Michael Strong, the current president of the Canadian Institutes of Health Research. Prior to joining CIHR, Dr. Strong was Dean of the School of Medicine and Dentistry and a distinguished professor at Western University. Dr. Strong earned his degree in medicine at Queen's University, followed by neurology training at Western University, and completed postgraduate studies at the Laboratory of Central Nervous System Studies at the National Institutes of Health in Bethesda, Maryland. Dr. Strong's research interests focused on understanding the neurodegenerative disease ALS. One of his lab's focuses was defining the role of alterations in tau protein metabolism in the frontotemporal syndromes of ALS and in traumatic brain injury. We at BEaTS Research Radio are delighted to host today Dr. Strong on our milestone 50th episode, and coincidentally, this year marking the 20th anniversary of the founding of CIHR. Welcome, and thank you for joining us, Dr. Strong.
Well, thanks, and congratulations on getting to 50 episodes. That's terrific.
Thank you. Thank you. Thank you so much. We like to start by asking our guests about the early career journey. What contributed to setting them onto the path that led them to where they are today? Can you tell us, Dr. Strong, a little bit of your journey from medical school to heading the preeminent health research funding agency in the country?
I'll be happy to. It's certainly not a straight line, that's for sure. In between
Never is.
Any of it. No, not at all. You know, it's nice to reflect on this even a number of times. And actually, this isn't the time this week that I'll be doing this. You know, it's all about the people that you've met along the careers, you know, people who have helped mentor you, even if they don't know that they were mentoring you at the time. So these are people who have influences on your careers. And I have to say, it really started for me just even before I actually graduated from medical school, because we had the opportunity to do an elective. And Henry Dinsdale, who was the chair of neurology at that time, I had approached him about going to Europe. And just, you know, out of the way, you know, things kind of work out the way they do, you just never really know. I ended up in Denmark, and I ended up working in the neuromuscular clinic and seeing patient after patient with Lou Gehrig's or ALS at play. I'd seen one case as a trainee before that, and was told I'd probably never see another case in my life. So of course my first thought was, gee, there goes Denmark. This shouldn't be happening for it all. But it got me interested in the disease. Why would it be so prevalent? I learned what a referral clinic was, right? So that was a pretty straightforward one. But when I got to do my neurology training program, Arthur Henson was there, and Arthur really established the ALS Society of Canada. It was one of two really North American leaders in establishing, you know, team-based care for ALS patients. And so we were seeing many, many patients coming through. And it was really Arthur who, you know, really moved me towards saying, you know, you really should be looking at a career as an academic neurologist. I remember that I'd gone into it thinking I'd really be a very good community neurologist. I was interested in maybe going to Peterborough or Barrie, that sort of an area. It was Arthur who said, you really need to look outside the box. And just at about that time, Carleton Gajdusek had come up to lecture, and Carleton had really just won the Nobel Prize for the discovery of Kuru. And Carleton spoke about Kuru and a whole bunch of other things, which I had really no interest in whatsoever. But he did talk about the hyperendemic focus of ALS that they'd been looking at in the Western Pacific. And he showed some features of the pathology that I'd been able to see because I was on my pathology rotation, and I was seeing many of the patients that I'd seen clinically for it. Now, again, it turned out that what I was looking at on the electron microscope was another artifact. It took me a year or two to figure that all out. But it was enough to really tweak my interest, and off I went to my postdoc at the NIH with Ralph Garudo, and went for 1 year thinking, oh, you know, well, let's just get a little bit of a flavor of this. Stayed for 3 years, and it was clearly, you know, it changed the course of where I was going to be, and really firmly implanted the idea of disease-based research. And like NIH at that time, and still was on the intramural programs, was the area where you went to do research on the chance that, you know, you really high risk research that was likely to fail, but when it didn't fail, had huge impacts. That was always the philosophy that was there. So it really bred in me a kind of philosophy that research is really something that's incredibly exciting, that you take risks to understand what a fundamental of a biology is, and that you're going to be wrong. You know, it's just the nature of the beast. But sometimes you're going to be right. And sometimes when you're right, it changes the way that you think about things. And so I did 3 years of that and then came back to Canada and I came back to Western where I, as you said, had done my fellowship training or as a clinician scientist. And at that time was supported by the MRC for 5 years with salary support. It was absolute seminal to me because it allowed me the time away from the clinic to be in the lab and to really get established. And at that time we were doing cell cultures and trying to understand in dissociated spinal motor neurons, a technique that we developed while I was at the NIH, how it was that individual cells had this susceptibility for it. Make a long story short, I just over the next 25 years, 30 almost now, I've been able to continue to maintain and grow that lab, but completely changed. When I came back, my work was really very much in developmental neurotoxicology. So, you know, looking at trace metal toxicology. And if you had asked me then, you know, PCR had just started to come out. We were still spooling DNA. You know, we were still counting cells by hands with a small cell counter. You know, sort of things that you guys nowadays, when, you know, the kids nowadays look at and kind of go, really, I saw that at the Smithsonian. I didn't realize people actually did that. But those were how we learned, right? The basic techniques that were in play. And here I am now, we're doing work in RNA stability, the determinants of it, looking at how tau metabolism interacts with other major proteins synergistically to lead to a disease induction. And then how does that link back to what we're seeing in our clinical phenomenology? So, you know, all of that pathway has in some senses always being at the right place at the right time with the right mentors or the right people saying, do you really want to, you know, do you want to go there? Why don't you think about this? Maybe go down that pathway and then being open and receptive to it. So it's been a great pathway to get to where I am now.
Yeah, this is definitely one of the hallmarks of a science career. It's the little nudge, it's the little question that then takes you off into years long worth of you know, discovery and learning, right?
Well, I have to tell you, you know, if we go back just even to one, you know, I'd been back in the ALS clinic for maybe a year or two, and we were seeing a lot of patients with pseudobulbar affect, which is the inability to control your emotional output, so tearfulness, laughter. And you can imagine how inappropriate that would be. You know, I was seeing people who were, you know, pastors and ministers and would start to laugh in the middle of a sermon. That's the type of thing that you would see. And I was working on trying to figure out how we could treat that pharmacologically for it, and it was getting nowhere, like really nowhere at all. And then one night, my wife and I and some friends were over at a colleague's who happened to be a neuropsychiatrist. And Peter and I were outside, you know, had a nice little glass of scotch with us. I had a cigar, because at that time, yeah, that's, you know, cigars were a thing of that day for it. And I said, you know, Peter, I can't treat this. I have no idea what's happening with it. And he sort of said, well, why don't you try this drug combination and see how it works? So the next day I tried it. And for 25 years after that, it was the drug combination that I used because it worked immediately, overnight. The symptoms evaporated for it. But the problem was, right, was that there were no receptors on motor neurons. There were no benzodiazepine receptors. There were no receptors where dizipramine would actually have an effect on motor neurons. And that's what led us to kind of standing back and kind of going, wait a minute, this doesn't make sense. These drugs should not have worked. So we must have another cell population that is affected. And just at about that time, the frontotemporal dementias were starting to be better described. And they had these features of dysexecutive function, pseudobulbar affect. And that's all she wrote. 25 years later, we have international criteria now. They're called the strong criteria for diagnosing this. We understand the underlying biology. And it was one night with a colleague simply turning to him and saying, I can't treat this. And thoughts started the whole thing.
Well, that's again reassuring to know that it's these like little conversations that you have with colleagues, and sometimes even folks that are not in at all in your space and research field, that trigger thoughts and trigger ideas that you never know where it could lead. That's what investigative biology and learning is all about.
Yeah, I agree.
Now, with respect, let's turn the corner here from the basic lab research and the
Mhm.
pharmacological research into your current role within CIHR as the president of CIHR. Now, as a quick primer for our science trainees and early career audience, what do you see as the main functions and goals of the agency, of the CIHR agency?
Yeah, that's a terrific question in there. So the CIHR, is the only federally mandated agency whose responsibility it is to conduct health research to improve health outcomes in Canadians. It involves knowledge generation, it involves knowledge translation, it involves training the next generation of researchers coming forward. And it has a very broad mandate in the sense that, it's all areas of health outcomes part. And that's what we do. It, the CIHR Act, which created us in the year 2000, is crisp. And even as we're completing and getting ready to bring forward a new strategic plan for that, you don't really touch the original scribing of why does this exist. So it is an agency. It reports directly to cabinet through the Minister of Health. So very close relationship with the portfolio. And so it's meant to be responsive to issues that are arising, not only in real time, but also in looking at issues of long-term disease consequences and how do we actually start to address those. Everything from very fundamental cellular, molecular biology, all the way through to policy and knowledge implementation or knowledge translation for it. So it's a huge mandate and at the same time, very focused.
Yeah, we're privileged here in Canada to be able to have an agency like that supports the research community from, like you say, the most fundamental basic questions all the way to the top-down policy making decisions, and to have it all at least congregate within the halls of CIHR is a fantastic setup. Now, this year is unlike any other year, and to reflect a little bit on 2020 with respect to our present COVID-19 pandemic, what did you see as the priority roles for CIHR? And to project a little bit also in the future, what ideas are forming with regards to supporting the science and the community members that are working towards COVID-19 crisis responses?
Yeah, it's a terrific question, and it has so many different facets to it, right? And so maybe I'll step back to the very beginning of all of this, right? So CIHR was there at the very beginning of this. We have quite an international outreach, particularly on the infectious diseases side. We have linkage through to WHO, to GLOPADAR, which is an agency that looks at infectious diseases worldwide. And so when this was really starting to percolate, become obvious that something was happening in January, we were already involved on the international front to begin to look at what the major research questions would be, even before it had landed on Canadian soil. And then we're highly represented and very much in part of the WHO roadmap that was sent up, or the blueprint for research that was developed in early February. So that within days of that being announced, we had announced a first research competition to try and start to focus on that. So it was actually the first one in the world to be done that was focused on the COVID-19, but broadly, without sort of saying, yes, we want you to begin the vaccine development, or we want you to, right? Ours was just turning to the Canadian research population and saying, listen, we need help here. Let's start to move forward on this. And at about that time, it was starting to become evident that this was going to become an issue for Canadian soil, that we were going to cases that this was going to become a pandemic. Remember, it had not declared that by WHO. Our government was already looking at it. And we got clear marching orders that as this was starting to come along from the Public Health Agency, the magnitude of all of this, that we would direct our resources towards a whole of government response to the pandemic as it was coming down the pike and then as it was evolved for it, which really meant setting aside virtually everything else that you do and directing all of your resources and time towards dealing with this. And so for us at the CIHR, because we are a research agency, that really meant focusing our research programs, getting everything in line to try and begin to develop what the response would be for this. And because we had the blueprint from the WHO and had been instrumental in helping to develop it, we already knew the major areas. Now, the difference between now and then was back in those days, we were kind of figuring, yeah, this thing's going to be done come July, June or July. Hindsight is always so crystal clear, right? And so we thought this was a transient period of time where we'd have to redirect our resources, then we could get back in to the stream of what we needed to be doing. But quite clearly, it became an issue that was going to be much longer. Clearly, it remains that way for it. And so as we've been doing the research and working with the government to provide timely answers, but also to work on some of the more fundamental aspects of it, we've been directing research programs that have had focus on development of vaccines. You hear about the world ones, but Canadians are actually world leaders in development of these vaccines. It's been tremendous work. We're also world leaders in understanding the multi-systems component of this. So you think about, in our world, you think about the cardiac effects of this and the receptor densities, and why is that? Why is it that there's a cardiomyopathy? In the renal side, where the second highest density of ACE2 receptors is, what's the long-term consequences? Why do you have neurological consequences to this and you don't have any receptors up there? Right. So there's a lot of work that's being, but then you also need to ask questions around, what about the societal implications of this? This has really laid bare the inequity in our populations worldwide, it's not just here, right? So if you look at what's happening in low and middle income countries, right, where we were moving forward very nicely on reducing poverty, for those curves have now inflected because resources have been moved over for that. And as that has happened, we're seeing increases in HIV, TB, malaria, and such. So, Canadians have always been very active on an international front for research. Well, this is an area that affects not just internationally, but ourselves, indigent populations in Canada, underserviced populations, people at risk of opioids, mental health disorders. You look at all of that, and it brings together all of the agencies. It brings SHRC, it brings NSERC to the table, it brings us, it brings NRC. And so one of the really reassuring parts of all of this is that you have seen a coalescence of research activities across the government, where, in fact, when you really look at what's being done, we're covering so many different things. I have to say, it was interesting, and partway through the summertime, the National Institutes of Health came forward with their 5 major strategies for battling the COVID-19 component to that, and we all read it with great interest. But we were already doing it. We're just so typically Canadian, right? That we kind of stand back and go, yeah, don't worry about it. We're getting it done. I didn't need to tell you about that. But they're there. We were doing it. And so, I think when all is said and done and we get to look at this in a post hoc manner, which will be done, and it will be done critically because we need to do better as we go forward with that. I think one of the pieces that we will see coming out of all of this is that, it was a very cohesive, very early, very aggressive response. And the research wasn't just CIHR, it was everybody coming to the table. CIHR really operationalizing it and bringing it forward. Now, as we come through the pandemic on the other side, right, most of us are now feeling somewhat comfortable that we're sitting in a state of, right, the hyperacute phase is over, right? Now, clearly it's not over everywhere, but the curves of the second are starting to flatten a little bit. We have vaccines that are more than just on the horizon. They are reality coming forward. Lots of discussion around those in play. So we need to start thinking about what steady state is going to look like and what the residual is going to look like afterwards, whether it be long COVID or the immunological complications or so on. We could go on for a long period of time about that. But I would argue that one of the most significant things that we're going to have to deal with is the destruction of the ecosystem of health research and the inequities that we've got that became really apparent very fast. So what do I mean about the ecosystem of research, right? So it's wonderful to be the CIHR to say, yes, we're the federal agency that's responsible for this. But by God, we're not alone, right? So if you look at the funding of health research in this country, it's charities, it's philanthropic organizations, it is the universities playing a major sector. We don't play, as a researcher, I still have my lab running full tilt. I don't pay rent, I don't pay square footage, I don't pay for electricity, I don't pay for heat. Indirectly, do I? Through the indirects, they're coming from my grants and otherwise? Sure, but it's not coming out of my grants. And so when you think about that entire ecosystem that allows us to do research, charities have been hammered through this. There are some that are going to disappear and we'll never see again. And I can tell you, as somebody whose career was built through the ALS Society, the Muscular Dystrophy, European, right, and CIHR from time to time, right, in it. In the absence of that, I wouldn't have had a career. I wouldn't be here today, for certain. So we're going to have to figure out how we get through this next couple of years as the system rebuilds itself, but is almost, it's not lethal, but boy, it's a wounded beast right now. And we have to fix that. The second area are the inequities, right? And we've done a lot of work to try and address those across all agencies, and particularly women in science, right? But it's not just the women in science, it's single parents. It's people who are taking care of their parents or elderly at home. It's people who have little children that they've had to start teaching at home, right? With this, who are no longer able to contribute to the extent they would like to on the research. And we know that's disproportionate to women. You just have to look at the data with regards to publication rates. Men have gone up, women have gone down. So we're trying to balance that on our grants. And that's got to be a fundamental lesson for us as we go forward and reestablish this ecosystem, is that these inequities have to be dealt with. And we couldn't have had a clearer signal than right now to do it. It's a fascinating time.
Yeah, this is definitely one of the key points about 2020 is all the lessons that we get to learn from this, and a magnifying glass on some of the weaker aspects that that existed in the system that are now up to the community and the agencies and every community member to readjust and basically advance and find ways to progress in a better way, right? To build back in a way that is more beneficial to the health research community that we all exist in.
Oh, absolutely.
Now, keeping our focus on the future, other than the COVID-19 priorities, what are some of the future areas of interest for CIHR to expand its role in advancing science and its community members?
Yeah, that's also a great question. It's timely, right? Because we spent the last 2 years developing our strategic plan going forward. And our strategic plan really is a generational plan. It'll get formally brought out in February, along with an action plan that speaks to how we will begin to implement this. So the first block of it is a 10-year block. But what we've done is we've set our vision out 30 years and said, look, as the CIHR with the mandate that we've got in hand, let's really reach out there. And what we heard from the Canadian population, from researchers, from the academic institutes and such was, we need to deal with the health inequities in this country. For a country of this nature to have them. And they are, we often think about them as geographic, north-south. We think about them as indigenous populations versus non. We have them as well in the core of our major cities, right? So why is it that in a country such as this, that we have such inequities? And those start at the fundamental basis of science. They start at understanding population health. They start at understanding knowledge translation and how it differs amongst different communities to bring an intervention. Global warming starts to play into that, right? Because as things get warmer in the north, we're seeing diseases recur that you wouldn't predicted. As immigration increases, we need to be thinking about things like TB and so on, and how that all moves forward with us. So, this concept of thinking about health equity and equitable health outcomes is a very broad reaching one, and really is what our strategic plan speaks about. So then you have to place that into the context of, right, but we're also scientists, and there isn't a single one sitting around this table right now who can tell you what science is going to be, in my own lab 2 months from now, let alone 10 years from now, right? Because it's evolving that fast. Think of the clip at which we're learning. And so you can't put barriers around what it is that research should look like, but you want to try and be able to say, but collectively we want to be here. So what does that look like? So for us, and I've said this to everybody who would ever listen to me, right, along this, is that I am an absolute believer in science for the sake of really good science. Canadians have always had a tremendous history of this. We do so much with so little. You know, I talk to my colleagues south of the border at the NIH with regards to funding, and they kind of go, yeah, really? You just described a burn rate for a week with your budget, right? Yeah, but you know what? Look at what we do, and look at how focused we are, and look at how many are at the table to help us do this. So I don't buy that. Could we have more funding? Absolutely. We could always do with more, but we would do with more people then and a broader base as well for it. So I do think there's a need for the individual investigators, you know, who have just the really great ideas. It's that chaos at the margins concept. Let it run, see what happens for it. But eventually, some of that has to start to coalesce into what are we trying to achieve. And that's the conversation that we'll be having as we go forward with all of this. So you can look at any sphere, right? And we think of it in the terms of the life cycle of an individual, you know, pre-embryonic all the way through. And really, if you're thinking about epigenetics, you're thinking about the environmental influences that are a generation before you're even a glint in somebody's eye, right? So you take all of that to the fact that we have a population of octogenarians, centurions that are going to be, right, over 100, mostly women, right? And you look at what's happening in Japan right now, and you see that they're leading that curve in there. What do we know about the pharmacology of anything in a 100-year-old, right? It's not the same. And what are the predictors of disease in your 6th, 7th decade of life that you set out on in your 2nd decade of life, right? And so all of those things, as we start to think about life as truly a life cycle, right? Our research has to touch on all of that. And it's so fundamental. You know, I see myself as I am a pillar 1 researcher. You know, I fundamentally work at mechanisms of RNA metabolism and protein turnover. Terrific. But at the end of the day, to what end, right? And where is it going to go? Well, that end may be that I'm one piece of a much larger microcosm that is actually trying to understand how degenerative diseases of aging are impacted on through the cumulative effect of insults over a lifetime, right, for it. So, you know, I don't have to be the one to change the world, but I got to be the one who's putting a piece to it. And so if you've noticed, I haven't really answered your question all that well around what is the, you know, what might be some of the fundamental ones in there, because I don't think there are fundamental ones. I think it is the science that we need to do to understand and be prepared to answer those fundamental questions. And how do all of these intersect? So it doesn't matter to me whether it's aging, dementia, heart disease, arthritis, just the list goes on, right? And so I think it's been one of the beauties and one of the real strengths of Canada that we are in a place to do that, and that we have such a breadth of training. We'll get back to it, you know, as we get through all of this. But that really is kind of where I see us heading.
Yeah, no, that's absolutely the case, and the future of science is definitely multidisciplinary research and group collaborative efforts to tackle these issues from as many angles as we can, from the as you said, the most fundamental biology aspects all the way to the translational and the bedside clinical patient outcomes that at the end of the day we all aim to improve and provide to our patients.
Absolutely.
Our last question for today is, as usual, we like to ask our guests for some words of wisdom or advice to provide to early career scientists, whether they be trainees or investigators, in order to achieve success. With you, Dr. Strong, I'd say achieve success with CIHR or just generally advance their careers.
Yeah, you know what? It's I've been thinking about that as we've been going through all of this, right? And the answer hasn't changed much over the course of years for me with that. And my colleagues that have enjoyed success, right? And I would say I've enjoyed success. I've had a terrific career. I've loved what I've done. And that's the key, right? To it is that this is a tough career. It doesn't matter whether you're coming at it from a PhD side of the equation. It doesn't matter if you're an MD who's doing additional research, if you're a health professional picking it up. It's competitive. We're tough on each other. We expect extremely high standards for it. And getting funding isn't always easy. I know many times I thought I'm just going to hang it up and play with my shingle and get back to work in the clinic and just call it a day for it. And I always had the right mentors, the right people, the right colleagues who talked about, you know, what a privileged life we have as researchers, right? We get to ask questions that nobody else does. We get to learn methodologies and approach questions in ways that nobody else could ever imagine. We have colleagues around the world who think differently and challenge us on a daily basis, right, about our results and make sure that they're correct and such. So if you don't love this job, if you don't love doing science, I don't care how fascinating your question is going to be, you're not going to enjoy the life that goes with it. And so I think one of the words of advice that I really have, and there are only two major ones, but the first one is really, you've got to find your niche in this research world that you can see yourself doing for years to come, with enough comfort in that area to know that you're constantly going to be changing. You know, the people that I've known who got into trouble over years in their research programs didn't really evolve their question. And that's so important, but you have to have that security. The second piece is that don't do this road alone, right? There's no way to do it alone. It's your friends, your colleagues, it's your family, it's your, you know, if you have a partner, if you have a spouse, they're going to be part of this from day one. You're going to come home, you know, depressed that your gels just like ran the wrong direction. Like, I've done that, right? This guy just wasted a month of work on this gel. I happened to put the electrodes on backwards. You know, there's stories that you could write about. You know, those are the sort of things you need to be able to have somebody you can say, listen, you know, I just need to talk to somebody. I've had one of these horrid days. Or, you know, that you get a grant review back and my God, you'd think you'd just written it in a language that nobody could understand. So that network around you, those friends, those colleagues, I can tell you some of my best friends in the field, the ones that I will call with scientific problems and that I'm still working with, are people I met 30 years ago, right? People that I was, you know, we were, again, you know, PCR hadn't been invented. We were sitting around and debating, can you believe this? Debating about whether you needed proteomics or you needed the RNA people or the DNA folks for it. Those are kind of healthy debates, and they led to friendships, you know, 30 years later that we still have. So I would say those are the two biggest lessons in this. You have to love what you do. You have to keep that over your lifetime, and you have to share it with somebody.
These are excellent and true words of wisdom, much appreciated, Dr. Strong. This concludes our interview. Once again, I'd like to thank our guest today, Dr. Michael Strong, for the informative and fascinating talk with us regarding the role of CIHR and advancing health research in the country. On behalf of our director, Emilio Alarcon, and the whole BEaTS Research Radio team, thank you all for tuning in. Don't forget to subscribe to our YouTube and podcast channels, and we'll see you again soon with another Science for the Community episode. Until then, stay safe and heart healthy.